Hi everyone, this is the continuation of the skeletal muscle lecture, and we're going to actually continue on at the end of this lecture and talk about smooth muscle. So, to pick up from where we left off in lecture, excitation is the first step in muscle contraction, and that's where you have to somehow transmit the action potential that travels down the motor neuron and make it so that it gets to the sarcolemma. And that's what the neuromuscular junction is all about. Excitation-contraction coupling refers to the transference of an action potential on the sarcolemma into the contraction of the myofilaments, and as we know, that is because the action potential on the sarcolemma contributes to the release of calcium from the sarcoplasmic reticulum. And contraction or sliding filament theory states that the thick and thin filaments are going to slide past each other rather than having the actual physical protein shorten. And this is contingent upon that calcium being bound as well as some stuff involving ATP. And then, of course, you don't want to contract and then never be able to relax. You don't want to have a one-shot contraction, so relaxation is just as important to understand conceptually in this context, because as we move and use our skeletal muscles throughout our lives, we are repeatedly contracting and relaxing them. So, what I'm going to do is annotate this particular drawing with some stuff from lecture as I walk you through it. So this red arrow represents the action potential coming down the axon to the synaptic knob at the end of the motor neuron. Remember that the action potential reaching that point opens voltage-gated calcium channels in this area, and the influx of calcium into the synaptic knob results in exocytosis of synaptic vesicles that are full of acetylcholine. So acetylcholine in this case is the neurotransmitter that is going to diffuse across the synaptic cleft and bind to the numerous acetylcholine receptors on the junctional folds in the motor end plate. So just to give us some anatomy, this whole thing that I'm bracketing right here is motor end plate. And each of these little foldy guys, there's one, there's one, there's one. These are the junctional folds. And the folds are there to increase the available membrane surface area to put receptors on. So the more receptors you have at a given spot of membrane, the more sensitive that piece of membrane will be to whatever thing they're designed to bind. So in this case, the thing that they're designed to bind is acetylcholine. And that is the neurotransmitter that is the ligand for this receptor, which is a ligand-gated ion channel. So if you watch my video about membrane potentials and development of charge differences across a membrane, I discuss both ligand and voltage-gated channels. Ligand-gated channels are ones that are only open when their favorite chemical is bound to them, whereas voltage-gated channels are channels that are only open at their favorite membrane potential. So that's the difference. So when you have 2-acetylcholine bound to the acetylcholine receptor, that channel is now open and ions can begin diffusing down their respective gradients, which is going to depolarize the sarcolemma. So that's how you get an action potential in a neuron to transfer over into an action potential in the sarcolemma. So that's how you get muscle excitation from nerve excitation. Now, that action potential is going to travel down the nearest T-tubule, and because we know that T-tubules are flanked by terminal cisternae of the sarcoplasmic reticulum, forming a triad, let's label that. That's going to let the calcium out because there's little voltage-sensitive doors in the sarcoplasmic reticulum here that pop open when the voltage changes, and then they pop back shut. So that's how the calcium gets out. We'll talk about how it gets back in a little bit later. So we know that once calcium binds to the troponin complex, that's going to cause a conformational change in tropomyosin that moves tropomyosin out of the way, and now you can see the active sites of G-actin are revealed. G-actin active site. And an active site is just a place on a protein that is designed to bind to something else. So, In this case, the active site is supposed to bind to myosin. So now we need to talk about what the preconditions for myosin being able to bind to actin are, because we've established what the preconditions for the revealing of the binding site is. So this is pretty close to the drawing that I drew in class. So here we have F-actin with revealed binding sites, because calcium is bound to the troponin complex. Myosin has to hydrolyze ATP. So remember, enzymatic hydrolysis is taking something and breaking it down with water. And once it's hydrolyzed the ATP and released a little burst of energy to do work, it can move into the cocked position. That's cocked as in cocking a pistol. That's what it is referring to or analogizing. And that is the position in which it's ready to bind and form a cross bridge with the actin. So that's what you're seeing here. So a cross bridge is the bind that is formed between myosin heads and one subunit of G-actin. So once that occurs, and I want to point out that the ADP and inorganic phosphate has been left out of this particular picture so that we can label the cross bridge, but it really doesn't leave the actin head until the power stroke. So in one movement, the actin head pulls backward while releasing ADP and inorganic phosphate. And that moves the actin filament this way relative to its initial position. So during a muscle contraction, you have lots and lots of myosin heads on the thick filament doing this many, many times to either shorten the muscle or maintain tension against a load. So load, for example, meaning, let's say I hand you a cinder block and I say, hold that, I'll be right back, and then I leave you alone with the cinder block. You're not changing the length of your muscle or shortening your sarcomeres, you are just resisting the gravity and weight of the cinder block by maintaining tension in your arm muscles. And hopefully for you, at some point I'll come back and take the cinder block away. So, in order for the myosin to let go of the actin, a new ATP has to come along and bind to the myosin head. If you don't have any ATP available to bind to the myosin head, the cross bridge will remain in place. So cells that are dead can't make ATP. So if a cell runs out of ATP, whatever cross bridges it has formed currently, it cannot undo because there's no available ATP to accomplish the task. And that, my friends, is one aspect of how rigor mortis forms. So rigor mortis is the stiffness in the muscles that occurs after death. And that's because once you die, you can't supply oxygen to your skeletal muscles, and they can't make ATP, which means that whatever cross bridges you have going on at the moment of your death, or near that anyway, are going to stay there until your lysosomes in your cells bust open and spill enzymes onto your muscle tissue that begins to break it down. There's another aspect of rigor mortis that we'll get to in a minute when we talk about relaxation. So this is the cross-bridge cycle, and it happens over and over again with many myosin heads forming and breaking cross bridges with many G-actins. So we've boiled it down to one pair here, but in an active sarcomere, this is happening many, many times at the same time. Okay, so what if you want to stop contracting? I just told you that if you want to stop contracting, you have to have enough ATP, so even relaxation of a muscle is not energetically free. Which is counterintuitive. You would think that uncontracting a muscle is relaxing and just letting go, but in fact, at the cellular level, you need ATP and you need to spend it to relax your muscle just as you need it to contract your muscle, so neither are free. First step, though, is stop sending signals from the central nervous system to contract. And that means that calcium is not entering the synaptic knob, and that means that vesicles of acetylcholine are not being exocytosed into the synaptic cleft. But that doesn't mean that there isn't acetylcholine left over in the synaptic cleft from the last exocytosis. So you have to deal with that stuff as well. So one thing that you need is something called acetylcholinesterase, And acetylcholinesterase, we abbreviate it ACE, which is funny, it is going to come on and break an ester group off of acetylcholine, rendering it unable to bind to the receptor. And then it's going to carry that broken down acetylcholine back to the synaptic knob so that it can be reassembled and put back into vesicles. Because acetylcholine is an expensive molecule to make, so instead of just tossing it after one use, you can continually exocytose and then recycle it with the help of acetylcholine. But regardless, if you want to stop contracting, you have to get the extra acetylcholine that's left over in here back into this knob. Otherwise, the signal to contract will be prolonged. Another component of relaxation that costs ATP is the return of calcium to the sarcoplasmic reticulum. So this doesn't happen by diffusion. It happens due to an ion pump called the PDB-101 calcium pump. So that's spending ATP to move calcium from the sarcomeres back into the sarcoplasmic reticulum. So costs ATP. So again, not energetically free. You've got to get that calcium back into the sarcoplasmic reticulum, otherwise it's going to keep binding to troponin and keep resulting in the formation of cross bridges. So once calcium moves off of troponin, tropomyosin moves back to cover the binding sites as well. So let's talk about what happens when something in these processes goes wrong or is interfered with by a chemical. So some pesticides, and I'm asterisking this some, because it's unreasonable to assume that all pesticides work the same way. They totally don't. So some pesticides contain cholinesterase inhibitors that bind to acetylcholinesterase and prevent it from degrading acetylcholine. So that results in spastic paralysis, which is muscle spasms and uncontrolled contraction, and that can result in suffocation if the muscles of inspiration, such as the diaphragm, are affected by this pathophysiology. Botulism is a disease caused by the bacterium Clostridium botulinum. The Clostridium genus of bacteria is particularly nasty. They like to form spores and hide in places you wouldn't expect. So this releases botulinum toxin, which blocks acetylcholine release, so you don't get exocytosis of vesicles in the first place. And that inhibits the generation of an action potential at the neuromuscular junction. So this results in flaccid paralysis, which is just a type of paralysis that's different from spastic. Another one of the Clostridium nasties is Clostridium tetani, and this is responsible for tetanus, which is sometimes called lockjaw because the spastic paralysis caused by tetanus tends to begin in the face and neck. So tetanus works a little differently, and it works via a mechanism we haven't really discussed yet, and that's that resting muscle tone, so a state of partial contraction in your skeletal muscle, is regulated by your central nervous system through neurons that act on the motor neuron to inhibit or enhance their activity. So glycine is an amino acid, and it's also an inhibitory neurotransmitter. And that is released in the spinal cord to keep the stimulation of muscles at a reasonable amount. If you block glycine from being released, you get overstimulation of muscles, which results in profound spastic paralysis, which is bad enough to kill you. So tetanus is dangerous. That's why we vaccinate for it all the time. So in the next slide, I will actually show you what that looks like. Ta-da! And here is an illustration by anatomy grandpa Frank Netter, who is a medical illustrator of great renown, and he has drawn for you a guy with a rictus of lockjaw. So you can see the platysma and the muscles of the neck are seized up, as are the muscles of the face and lower jaw, producing this grimace. So botulism is foodborne, so you can get botulism from eating food that has been canned or jarred improperly. But remember, this prevents release of acetylcholine, resulting in flaccid paralysis. So you get no acetylcholine release, which means you don't generate an action potential in the first place. Inhibitory neurotransmitters still work just fine. release of neurotransmitter at the neuromuscular junction is impaired. Tetanus, again, another Clostridium bacterium, but this one is tetani. This is soil-borne, or it can be found on rusty nails in your garage, so put on your shoes before you go to the garage. And that results in rigid or spastic paralysis. So here we have a synaptic knob that is abutting the synaptic knob of the motor neuron. So this neuron here is going to regulate this neuron here. So that keeps this neuron from stimulating this muscle inappropriately. And it does that by releasing glycine, which inhibits the release of neurotransmitter. If you block the release of glycine and GABA aminobutyric acid, which is what GABA stands for, you get overstimulation of muscles, which results in rigid paralysis. So that's the difference. Now, I mentioned resting muscle tone, but I haven't really told you what that means yet. So to talk about that, we need to discuss the length-tension relationship, which is what it sounds like. It's a relationship between the length of a sarcomere and the amount of tension it is able to generate based on that length. So we'll look at a graph of this for the next slide, but for now, let's just define some terms. So the amount of tension a muscle is able to generate depends on how long it is before it was stimulated. So if the muscle is overly contracted, weak contraction results because the thick filament tips are too close to the Z-disc and they don't really have any room to slide any further. Overly stretched sarcomeres have very little overlap, so there aren't very many cross bridges between the two, which means that there's not a lot of grip of myosin on actin. So in either case, you get weak contraction. Optimum resting length produces the greatest force when muscle contracts, and that's a constant state of partial contraction that we call muscle tone. So here is that graph that I promised. Now I want you to notice percent of maximum tension is unitless because it's just percent. And down here we have percent of resting sarcomere length. So halfway contracted is considered optimal resting length. So 100% of optimal resting length is about 50% contracted. If you have significant overlap, the distance between the thick filament and the Z-disc is very small, so there's not much more contracting this sarcomere is capable of doing. So at low sarcomere lengths, you get very little tension. Conversely, at high sarcomere lengths, you get excessively stretched sarcomeres, so there's like only one cross bridge here, which means that there's not a lot of grip of this thick filament on this thin filament. So the more overlap there is, up to a point, the more tension you can generate. So that creates this hump-shaped curve. So the next thing we need to discuss is recruitment. And recruitment refers to motor units. So remember, a motor unit from earlier in the PowerPoint is one motor neuron and all the muscle fibers within a muscle that are innervated by that neuron. So, what are we looking at here? The proportion of nerve fibers excited, so this is a cross-section of a spinal nerve, and the little yellow circles, or in this case the little white circles, these are individual nerve axons, so an implied motor unit. So this is one motor neuron. So down here we have the stimulus to the nerve, and this is just stimulus voltage in millivolts, but it's unitless here because it doesn't really matter. We're just looking at more or less. We also have a threshold, which is the minimum stimulus required to excite a neuron and therefore a muscle. So note, you can change the voltage to a neuron a little bit, so this is not zero and neither is this, but get zero tension generated because your stimulus magnitude has not reached threshold. So we call these subthreshold stimuli. Once you reach threshold, perhaps one neuron is excited, resulting in its motor unit being activated, generating a little bit of tension. So if you need to use your muscle in a way that doesn't generate the maximum possible contractility, like you're going to lift something but it's not very heavy, like a pencil, you're not going to recruit very many motor neurons because you don't need to. You only need to recruit one. Suprathreshold stimuli are these, and that's because they're above threshold. So up to a point, the larger the magnitude of a suprathreshold stimulus, the more motor neurons are excited and the more motor units are recruited, which results in an increase in tension. But at some point, you're going to be exciting all of the motor neurons innervating a muscle, which means that you are recruiting all of the motor units available in that muscle, and that is the maximum amount of contracting that that muscle can possibly do. So maximum contraction equals all motor units active and contracting. So this is called multiple motor unit summation, meaning the more force you need to generate, the more neurons are firing at the muscle, and the more motor units you are recruiting for whatever task you're doing. So it goes back to that tug-of-war analogy. More hands on the rope results in greater tension generated. So the more sarcomeres you have actively contracting, the more contraction you're going to be able to produce. However, it does max out. So notice that at point number 7, all of the neurons are excited. So even though the stimulus voltage at points number 8 and 9 is greater than that at 7, you can't recruit any more neurons or motor units, so the amount of tension remains the same. You max out on tension, can't make any more tension. Okay, so that brings us to the concept of twitch and Treppe. So depending on how many times you ping a muscle with an action potential, you're going to get different generations of force. So a myogram tells you how hard a muscle is contracting. So that's on the y-axis here. And then down here we have action potentials over time. So frequency equals number of things over time. And in this case, the number of things is action potentials. So if you ping the muscle with one action potential and then stop, it's going to generate some force and then relax. So at low frequencies, up to 10 stimuli per second, each stimulus produces an identical twitch response. So you're going to have twitch, and then twitch, and then twitch. At moderate frequencies, so between 10 and 20 stimuli per second, each twitch has time to recover, but not all the way back down. So this black line represents the recovery that this particular twitch was unable to do, because the next stimulus from the neuron arrived before it completely relaxed. So now we get summation, which is where you can increase tension generated in a stepwise fashion by stimulating the muscle again before the previous twitch has had time to come down from contraction. So this takes on a staircase-like shape, which we call Treppe. So that's what that means. So high frequency generates even more contraction. So we start to get temporal wave summation. And that's at 20 to 40 stimuli per second. So each stimulus arrives before the last one recovers. That's what that means. So Treppe is a result of temporal wave summation. So incomplete tetanus is sustained but fluttering contractions. So you're continually generating tension, but you can feel your muscle fluttering. So the maximum frequency stimulation possible is between 40 and 50 stimuli per second, but that's a lot. So normally, below 40 to 50 stimuli per second, you get sustained fluttering contractions. So think of a time when you had to hold something heavy for a long time and your muscle started to stutter while it was maintaining tension. You were experiencing this incomplete tetanus. So hold heavy thing for a long time. At the maximum frequency, so above 40, between 40 and 50, the muscle has no time to relax at all because the signals to contract are coming so fast over time. So the twitches fuse into a smooth, prolonged contraction, so you get this nice smooth increase, which plateaus once it's at maximum contractility. So this is called complete tetanus, and this is really rare in the body. So in most ordinary operating conditions, the body doesn't do complete tetanus. It's individual or Treppe. So we've talked about the preconditions for contraction and relaxation, and one of the things I mentioned was it's not energetically free to contract, nor is it energetically free to relax. So all muscle contraction depends on adenosine triphosphate. It's the energy dollar of the cell. It's the energy currency of the cell that the cell uses to spend and do work. But there's more than one way to make ATP. So ATP is sometimes made using oxygen via aerobic respiration, but this requires a continuous oxygen supply, and it produces water and carbon dioxide as waste. So these are ones you breathe out. So at low activity levels to resting, you do aerobic respiration with your muscles. Anaerobic fermentation occurs when you are really stressing the system and you are demanding ATP be produced faster than you can provide oxygen for the production of it. So there's a supply and demand issue here. But luckily for you, you can do the quick and dirty ATP production, which is where you can make ATP really fast, but not that much of it. So aerobic respiration produces 32 to 34 ATP per glucose, whereas anaerobic fermentation produces like 2. So lots less efficient, but lots more immediate. Unfortunately, it also produces lactic acid, which is a byproduct that can be harmful in large amounts. So what you're seeing here is somebody that's going to do a dead sprint from resting. So between resting and a little bit into their run, they're going to start out using aerobic respiration. As they continue to run, it's going to start to be the case that they can't breathe in and out fast enough to supply oxygen for the rate at which they're using ATP. So a system called the phosphagen system takes over, and that is stored ATP and taking phosphate off of a molecule called creatine phosphate. So we'll get to that in the next slide, but put a pin in that for now. After you've exhausted your phosphagen system substrates, you move on to the glycogen lactic acid system. So this is the anaerobic fermentation. So you're producing ATP really, really fast without oxygen, but less efficiently. And because it's less efficient, and because it has a nasty byproduct, the total amount of time that you're able to do this is reduced, because there's an upper limit to how much you can do. So once you stop running, so let's say you've sprinted for 60 seconds and now you're stopping, you're going to put your hands on your knees and pant for a little while before you're okay again. And that's you repaying your oxygen debt. So as you were sprinting, you were using your muscles faster than you could supply them with oxygen, and you have to pay that oxygen back once you're done. So you have to put oxygen back on myoglobin and re-up your oxygen supply in your muscles. So that's why you do that panting thing at the end. So as promised, here are the energy systems, not necessarily in order. So in the previous, we saw this was 1, this is 2, and this is 3. So the aerobic system has a low rate of ATP production, so this one is how fast. Capacity refers to how much ATP is produced. And then fuels used just describes which fuels are being used, either ATP itself or to generate ATP. So, aerobic respiration has a low rate of ATP production, and that's because it's slow because it requires a lot of steps. Conversely, glycolysis, which is another word for anaerobic fermentation, has a high rate because it has few steps. Basically, you just split sugar, use the product of that, and then the rest of it is lactic acid. So this has a very high rate of ATP production, but it doesn't produce very many ATP. Whereas aerobic produces lots. The phosphagen system is basically either using ATP that you've stored up or taking the phosphate off of creatine phosphate and using that phosphate to phosphorylate ADP. So you're making new ATP by phosphorylating ADP as you make it, turning it back. But you run out of these quickly. So that's the trade-off. You can either produce lots of ATP or produce ATP quickly, but you can't really do both within the same cell. So fatigue is the natural logical outcome of thinking about this. So what happens if you use your muscles so much that you just run out of stuff? You don't have any ATP anymore, or you can't supply oxygen anymore. Then you get fatigue. So these two people are probably recovering from fatigue after doing some sort of track and field event. So ouch, that doesn't look fun. So fatigue manifests as progressive muscle weakness and loss of contractility from prolonged use. So can't generate tension. As glycogen is consumed, remember glycogen is stored in skeletal muscle, and it's a sugar storage molecule. You run out of fuel to make ATP. And remember that the sarcolemma is maintained, its resting membrane potential is maintained by the sodium-potassium ATPase. So the sodium-potassium pumps that reset the membrane and make it excitable, if you run out of ATP, they don't work. So then you fail to maintain membrane potential and therefore lose excitability. So even if you exocytose acetylcholine at this neuron all day long or at the muscle all day long, it's not going to do anything because ionically it's incapable. Lactic acid lowers pH, inhibiting enzyme function. So if you're using enzymes to, say, use the phosphagen system, that's going to stop working. And accumulation of extracellular potassium lowers the membrane potential and excitability. So the more positive charge you have in the form of potassium sitting outside the cell, the less excitable that membrane is going to be. You can also run out of acetylcholine. So if your neuron is exocytosing and exocytosing and exocytosing acetylcholine lots of times in a row, it may not be able to keep up with the demand and it's going to run out. So all of these things contribute to fatigue and exhaustion, meaning your body says, okay, it's time for you to stop. There is a physiological reason why you can't go. Depending on how you set a skeletal muscle fiber up, which is to say what you put inside of it, you can optimize it for either slow oxidative or fast twitch. So remember that muscle fibers refers to a cell. So this is a modification that you can make to skeletal muscle cells to optimize their performance under certain circumstances. So this is a electron micrograph where different types of muscle cells have been stained different colors. So first we're going to divide them up into two groups, slow oxidative and fast glycolytic, also called slow twitch and fast twitch. Slow oxidative use oxygen to make ATP, so they have more mitochondria. lots more myoglobin, which remember is an oxygen storage molecule, and lots of capillaries because they need access to oxygen carried in blood. So because they have so many capillaries, they tend to be dark red. So these are adapted for aerobic respiration, and they are resistant to fatigue. So you can use them for a really long time without them ever running out of fuel. So this includes the soleus and the postural muscles of the back, so the ones you use to stand upright. And they have relatively slow twitches, so 100 milliseconds per twitch, which is pretty long in the time frame of muscles and nerves. Fast glycolytic or fast twitch fibers have lots of those enzymes for the phosphagen and glycogen lactic acid systems. They also have a sarcoplasmic reticulum, which is primed to release calcium more quickly through mechanisms that we won't discuss. Faster contractions are quicker twitches, so about 7.5 milliseconds per twitch compared to 100. That's quite a difference. And these include things like your extraocular eye muscles that you move your eyes around with, your gastrocnemius, which is in your calf. You can use this for jumping, and your biceps brachii, which is the muscle in your arm. So with regard to types of skeletal muscle fiber, I just told you about fast and slow twitch, but it's a little bit more complicated than that. So type 1 are red muscle fibers, rich in capillaries, rich in oxygen. So these are slow oxidative and slow twitch, and they have a really high resistance to fatigue. So in a muscle that was designed for endurance, you would expect to see more type 1 red ones. Splitting the difference are type 2A, so these have pink and red muscle fibers, and these are fast oxidative. So the systems in there are primed to still be oxidative, but to do it more quickly. They're sort of a middling between the two processes. Most human muscles are this, so they are mixed fibers with an intermediate resistance to fatigue, and that makes them sort of a general purpose muscle. White muscle fibers are fast glycolytic. They have fast twitch fibers, and these have low resistance to fatigue, but they can produce a lot of power very quickly. So these are muscles that you might use to sprint or perform short burst feats of strength, so throwing things, jumping, for example. Those would be times when you would want to use white muscle fiber. So that concludes our discussion of the physiology of skeletal muscle, and in the next portion of the presentation, we're going to move on to the sort of structural details of the muscle as an organ. All right, so moving right along, we are going to start now talking about the muscle as an organ. So remember from lab, if you were there and if you're my student, if not, welcome to this part of the lecture. Superficial fascia is synonymous with hypodermis. So that is the layer under the dermis where fat and adipose tissue is stored. So it's very vascular and very fatty. So if you get liposuction, this is the layer that your surgeon is trying to suck out of you. The deep fascia is found between adjacent muscles deep in the arm. So that's, or any limb. This just happens to be an arm, which is why I said that. So this is going to be dense irregular connective tissue around and between the adjacent muscles. Speaking of dense irregular connective tissue, here we have a list of the connective tissues of a compound muscle organ. So the muscle as an organ is called an organ because, remember, an organ is anything made of 2 or more tissue types. So greater than 2 or equal to 2 tissue types. So that's why we consider skeletal muscles to be organs. So surrounding the entire muscle, there is dense irregular connective tissue called the epimysium. So epi means superficial to, and mesium means muscle wrapping. And as the name would suggest, it surrounds the whole muscle. And this is often continuous with tendons. Remember that muscles are bundled into fascicles, so they are subdivided, and a fascicle is a collection of between 10 and 100 muscle cells. So the perimysium is the dense irregular connective tissue that surrounds the fascicles. You also have a connective tissue barrier between your adjacent muscle cells. So individual muscle fibers are divided from each other by a layer called the endomysium. So endo means inside of muscle, peri means around muscle. So endomysium separates individual muscle cells. And that begs the question, well, why would you want to do that? Why would you need individual muscle cells divided from each other? The answer is electrical insulation. So if we established from the fatigue slide that changing the ions in the extracellular fluid around a sarcolemma changes its permeability to other ions and changes its electrical properties, let's say you have fiber A and you have fiber B, and fiber B is in a different motor unit than fiber A. So fiber B is going to depolarize because it's been excited. And when it depolarizes and then repolarizes, for a short time, suddenly there's going to be a lot of potassium in the extracellular fluid between muscles A and B. We know that lots of potassium in the extracellular fluid decreases the likelihood of a sarcolemma to be excitable, so it decreases excitability. So what that means is if I have fired muscle B first, and I decide to fire muscle A second, I'm going to have a harder time doing so because I've created an ionic environment that is unfavorable for excitation. But you can solve this whole problem if you just put a barrier between these guys such that the extracellular fluid around each muscle cell is isolated from that of its neighbor. So it performs the same function as electrical isolation of wires, where you don't want electron streams crossing each other. That shorts your circuit out. Tendon is actually dense regular, so this bullet point should be moved this way. I apologize. And that's because you want those collagen fibers all running in parallel to resist particular unidirectional tension. So these attach muscles to bone. Therefore, tendons are considered to be part of the muscular system. An aponeurosis is sort of a broad, flat tendon. So you have one of these on the top of your head called your gallia aponeurotica. And you also have one on the anterior surface of your abdomen. Occasionally, tendons need a little bit of help if they're very workhorsey tendons that move a lot. And so these tendons may be enclosed in synovial tubes that help lubricate the passage of the tendon and isolate it from certain tissues. So here we have a picture of all of those linings. So you can see the tendon and the fascia are continuous with each other. Epimysium is on the surface of the muscle. Perimysium is around adjacent fascicles. And so here's the perimysium here, blown up. And then endomysium would be around an individual fiber. So speaking of tendons, this should say dense regular. So tendons attach muscle to bone. So these are elements of a muscular system. Ligaments attach bone to bone, so they are considered to be part of the skeletal system. So some individual differences. Some muscles have more than one point of origin or insertion, meaning that there's multiple tendons or tendinous inscriptions or different attachment styles, and we will look at these in pictures in a little bit. So naming of skeletal muscles is something that you guys are doing in the lab right now. You're learning the names and locations of muscles and where they attach to the body. And you can name muscles based on a variety of criteria. So one criterion is the direction of fibers. So rectus muscles have fibers that go straight up and down. Transverse fibers go in the transverse plane, and obliques are anything diagonal. You can also name a muscle after its location. So the tibialis anterior is the muscle that's literally on the front of your tibia. So its name describes where you found it. You can also name muscles according to relative size. So relative means relative to something else. So for example, in the glutes, you have your gluteus maximus, medius, and minimus. So big glute, medium glute, little glute. And you can also have names like brevis and longus, so short and long. Sometimes muscles are named for their number of origins, or heads. And these muscles have names like biceps, triceps, and quadriceps. So a common spelling error I see for these, for whatever reason, is students want to say it's called a bicept with a T. That's not a word. Don't use it. Ceps means head. Bi means 2. Tri means 3. Ceps means head. So 2 heads, 3 heads, and quadriceps, of course, means 4 heads. Now, here's the deal with these. You have a biceps femoris and a biceps brachii. So biceps femoris means 2 heads leg, and biceps brachii means 2 heads arm. If you, on an exam or on a practical, just write biceps and you don't tell me whether it's the leg or arm one, I'm not going to be able to give you full credit for that answer because all you've written is 2 heads in Latin, and well done you, but you haven't told me where the 2-head thing is. So you need to give me as much information as possible. Some muscles are named for their shape, and this is relative as well because of course most muscles aren't perfect shapes, but your deltoid is vaguely triangle shaped, which is also the Greek character delta. The trapezius is roughly trapezoidal, and the serratus has a serrated edge. Other examples of shape names are your rhomboideus, so that goes between the medial border of your scapula and your spine, creating a diamond or rhombus shape. So those names are named after the polygonal shape of the muscle. Some muscles are simply named for their origins and insertions. So an origin is going to pull the insertion towards it when the muscle contracts. So sternocleidomastoid is a good example. Sterno means sternum, right there. Cleido means clavicle, right here. And it inserts on the mastoid process of the temporal bone, right here. So the name literally tells you the origin and insertion. So if I ask you the insertion or the origin of the sternocleidomastoid, there is no excuse for you not remembering it. It's literally right there in the name. Muscles can also be named for their action, so what they do. So for example, you have a flexor carpi ulnaris. So it flexes the carpal bones on the ulnar side. So it's named for what it does. So again, these ones are named for their action, so if I ask you the action for something that is named its action, you should be able to interpolate the action from the name. So arrangement of fasciculi. So remember, fascicles are the bundles of muscle cells, between 10 and 100 muscle cells, surrounded by paramecium. And the way that you arrange fasciculi within a muscle can change the kind of jobs that the muscle can do. So there are 4 major patterns for this. There are parallel fascicles. So the sartorius is an example of one. Another example would be the gracilis. Fusiform is a muscle that has a broad belly and the tendons come to 2 points at the end. Pennate means flag-like, so these can be unipennate, where you have a tendon going up the side of the muscle and the muscle's belly or fibrous contractile part hangs off the side, or bipennate, so you can see there's a central line on this muscle and then the fibers run obliquely from that central line. Or multipennate, in the case of the deltoid, many pennae, and you can activate different ones of these depending on which of the actions of the deltoid you're doing. So are you extending or abducting your forearm or your arm? You can activate different pennae depending. And circular, which is what it sounds like. So the orbicularis oris is one of the muscles I'm using right now to change the shape of my mouth to produce speech. So organization of fasciculi affects power, range of motion, and speed of movement. So the longer a muscle fiber is, the greater its range of motion is because there's more of the muscle to contract or relax. The number of fibers in a muscle reflects the maximum amount of tension it can generate. Obviously, if you have more sarcomeres, you can generate more tension. If you've ever thought about muscular physiology and bodybuilding, that seems pretty clear, right? More fibers or bigger fibers equals more tension generated. Fascicular arrangement represents a compromise between strength and range of motion. So if you want to have a lot of fibers, you're going to have to give up a little bit of range of motion and vice versa. So now we're going to get into a little bit of applied biophysics. So in physics, a fulcrum is a fixed point about which something can rotate under different forces. So each bone is a lever, which is a rigid moving structure, and it's acted upon by a muscle to change the applied force. So applied force is also known as effort, and this is literal effort. So if I pick up something heavy, I'm making a strong effort to pick it up. And using the example of the heavy thing, effort is required to overcome a load. So if I want to pick a cinder block up off the ground, I'm going to have to apply a force that is equal to the mass of the object, and in fact is greater than the mass of the object because I'm going to have to move it. So the function of a lever is to change the direction of an applied force. It's to change the distance and speed of movement produced by an applied force, so I can lift things slowly or more quickly, and I'm going to need to apply force differently depending on what I want to do, and to change the effective strength of an applied force. So if I'm holding a load and then I put another load on top of it, to maintain the same position, I'm going to have to increase the applied force or effort that my muscle is doing. So if you're already holding a stack of books and I stick another book on top, you're going to need to generate more tension than you were previously to maintain the same position. So if you boil down a lever to a teeter-totter, or a seesaw, whichever you prefer, that's a style choice, what you get is something that looks like this, where there is a fulcrum that is fixed, it does not move. And then if effort equals resistance, you get a flat line, so no movement. If effort overcomes resistance, you get movement. So if the amount of effort I apply this way is more than the resistance, I'm going to get a deflection of my seesaw this way. If the resistance is greater than the effort, I'm going to get a deflection of my seesaw in this direction. So if you've ever been on a seesaw or seen one, this should make kind of intuitive sense. And the same components are true of a lever system in the case of muscles. So to spin out this concept, we've just talked about origin and insertion, and we've just talked about the lever system. So it makes sense that muscles primarily originate or insert on the skeleton, because if you have a muscle spanning two bones, you can change the relative position of those bones relative to each other. And the reason this says primarily is because, as you've seen from lab, especially in the case of things like the muscles of the face, some of them originate or insert on like skin of lips or modiolus of mouth, which isn't a bone, but rather a spot on the face or some skin. So there's a little asterisk next to primarily because that's not always true. So the origin is the fixed point of attachment, and in the case of limbs, it's usually proximal to the insertion. Insertion is the moving point of attachment, and it's generally true, not always, but oftentimes true, that when you flex a muscle, the origin is going to stay the same, and the insertion is going to get closer to the origin. So for example, here is the insertion of the biceps brachii on the radius. If I flex my biceps brachii, I'm going to move my radial tuberosity, closer to the humerus, which is the origins. So action is the movements produced by muscle contraction. So we have body movements like flexion, which is decreasing the angle of a joint, extension, which is increasing the angle of a joint, adduction, which is moving a limb from lateral position back to the midline, or abduction, which is the opposite. moving a limb from the midline away from the center. And these are described in terms of bone, joint, or region. So the actions of the muscles are named for the bones that they move. So for example, the action of the levator scapulae is to elevate the scapula. So it's going to move the scapula up. Group action describes the cases where muscles, instead of acting on their own, work in groups to maximize efficiency. And group action is the norm. It's very rare that muscles act completely independently of one another. They come in groups, and groups have different roles. So an agonist is a prime mover, and this produces a particular movement. So in the case of this example, flexes elbow, which means it's going to decrease this angle here. So right now we're at about 90 degrees. If I want to flex it more, I'm going to decrease the angle to maybe 45. So in this case, the prime mover is going to be the biceps brachii, because that's the one that straight up flexes the elbow. An antagonist is going to oppose the movement of a particular agonist. So in this case, the triceps brachii, which would be on the back if it hadn't been dissected away here, is going to oppose the action of the agonist. And the synergists are going to be smaller muscles that assist a larger agonist, either by cooperating to produce the movement or stabilizing a joint during the movement. So we call those fixators. So let's look at the example I just described. Here we have the biceps brachii. Oops, sorry, my bad, go back one. We have the biceps brachii, we have the brachialis, and we have the triceps brachii. So notice I'm being very precise about my language. This is the 2 heads arm. This is the 3 heads arm. So the biceps brachii is the prime mover. That's the agonist. The brachialis is a synergist. It's smaller and shorter, meaning it can't generate the same amount of tension as the biceps brachii, but it does help with elbow flexion. And the antagonist is the triceps brachii. So if this is contracting, it's going to pull the olecranon of the ulna towards the olecranon fossa, straightening your arm out. And that's the opposite of flexion, so that's extension. A fixator is a muscle that holds the scapula firmly in place during arm movement. So an example is the rhomboideus major. That's one of those ones that goes between the medial border of the scapula and the spine. So I've just drawn it right here, R. So it is possible to generate tension without movement, and it is possible to produce movement while maintaining the same tension. So there are different ways of using your muscles depending on what you need to do. So isometric contraction, iso means same, and metric means length. This is developing tension without changing length. So this is isometric. Isotonic muscle contraction comes in 2 flavors. You can develop tension while shortening. So if you're lifting something, you're going to shorten your biceps brachii to overcome this load and apply force upward. Or if you're going to lower the book back down in a controlled fashion, you're going to perform eccentric contraction, which is where you're developing tension by lengthening. And that concludes our discussion of skeletal muscle. So moving along, let's talk about other types of muscle. So smooth muscle is not striated and it's involuntary. So I can't choose to use this using higher cognition in my frontal lobe. And it doesn't have sarcomeres or Z-discs in the same way that cardiac or skeletal muscle does. And also, unlike cardiac and skeletal muscle, smooth muscle can divide and regenerate, so can fix itself. And that's lucky for us because the walls of hollow organs are primarily made of smooth muscle, and these are all over the place. So blood vessels, for example. Imagine if you could never fix your blood vessels, you'd be in trouble. The urinary bladder, the respiratory tract, the digestive tract, and the reproductive tracts all have smooth muscle in them. So the reason that the skeletal muscle system is considered to be an organ system, but the smooth muscle isn't is because the smooth muscle is a member of many organ systems, not just one. So here is what a smooth muscle looks like. This is a single cell. So A is relaxed, B is contracted. And these are fusiform, meaning they have a broad center and pointy ends. So these are between 3 and 200 microns long, depending on location, and 5 to 10 microns wide. They don't have any visible striations, sarcomeres, or Z-discs. Instead, they have thin filaments attached to dense bodies, here pictured in black, scattered throughout the sarcoplasm and on the sarcolemma. So that's where the thin filaments are anchored as opposed to a Z-disc. So I'm going to write not Z-disc. The sarcoplasmic reticulum is scanty with no T-tubule, so there are no invaginations of the sarcolemma deep into the cell because there don't need to be. And you don't need a developed sarcoplasmic reticulum because smooth muscle has lots of access to the extracellular fluid. So primarily, the calcium necessary for smooth muscle contraction comes from there. A little bit may come from the sarcoplasmic reticulum, but not much because it's much smaller. So, tying this back in with our skeletal system lecture, remember I mentioned that calcium in the blood is really tightly regulated. So, blood calcium ions filter out of the capillaries to form ECF, extracellular fluid, or tissue fluid calcium ions, and those are the ones that supply the calcium for smooth muscle contraction. If present, the nerve supply to these is autonomic, so this is an involuntary part of your nervous system. And unlike skeletal muscle, now we have two options for neurotransmitters to make this go. So smooth muscle can be excited by acetylcholine, just like skeletal muscle. It can also be excited by norepinephrine, aka adrenaline. And it depends on if you're in rest or digest mode or in fight or flight mode. So when we get to the autonomic nervous system, you'll learn more about that. But this is as much information as you need for now. So because there's two neurotransmitters that can act on this muscle, you get different effects in different locations. So for example, acetylcholine dilates your pupils because your pupils are controlled by smooth muscle, whereas acetylcholine constricts rather your pupils, adrenaline dilates them to let more light in to prepare you for action. Oops. So, stimulation of smooth muscle is involuntary and contracts without nerve stimulation necessarily. So remember I said nerve supply, if present, is autonomic. That's because some of these smooth muscle structures have pacemaker cells, which are cells that spontaneously depolarize and produce contraction without the input from the central nervous system. So that can happen. So one example of that is your enteric nervous system. So pacemaker cells in the GI tract are autorhythmic. They depolarize spontaneously. Other things that can stimulate smooth muscle contraction are hormones. So one great example of this is oxytocin. In addition to having a role in social and affiliative behavior, oxytocin is also a potent stimulator of smooth muscle. So some notable examples of oxytocin's action on smooth muscle, particularly in the context of human sexuality and reproduction, are oxytocin has roles in orgasm and climacteric in both the male and female sexual response, and it is a critical component of labor and delivery of a fetus. Carbon dioxide, which is a metabolic byproduct of cellular respiration, can also activate smooth muscle, as can changes in pH, being stretched. So bladder is a great example of this. There are stretch receptors in your bladder, and when they reach a certain critical mass of stretchedness, it causes your bladder muscle to contract, resulting in the feeling of needing to urinate. And also oxygen deficiency can cause smooth muscle action. So if you're not getting enough O2, you can blow open the smooth muscle in your bronchioles and your airway to let more air in. And continuing with our theme of this being different from skeletal muscle, autonomic nerve fibers have bead-like swellings called varicosities, and these contain synaptic vessels. So a varicosity might look like this, where there's an axon, and there's a varicosity, and there's another one, and there's another one. So the place where the vesicles are is not the end of the cell. So let's draw some neurotransmitter vesicles in there. So this stimulates multiple myocytes at diffuse junction. So if I excite this neuron, it can excite this myocyte and this one, and maybe one over here as well. So we're not going to get super in-depth into the contraction of smooth muscle because it's way more complicated than skeletal muscle, but I do want to give you a flyover view of how this works. So the calcium triggering contraction is extracellular, and that enters the cell either through voltage-gated channels or hormones or neurotransmitters or stretching. So there's a variety of mechanisms. We're not going to go into the individual physiology of each. Regardless, though, the fate of that calcium is to bind to not troponin, but calmodulin. So cal equals calcium. To modulate something is to regulate or control it. And the suffix in means protein. So calmodulin means calcium regulating protein. And I'll show you a schematic diagram of this in a minute. So what calmodulin does is activates a particular enzyme called myosin light chain kinase. We'll see it again in the next slide. And that hydrolyzes an ATP and then another one, which gives myosin the ability to perform the power stroke. So the first ATP is spent to activate the myosin. Second ATP is spent during the power stroke. So you do have thick and thin filaments in smooth muscles, they're just not arranged into sarcomeres. And what you get is the shortening of the entire cell in a twisting fashion, sort of like wringing out a washcloth. This also goes really slowly compared to skeletal muscle, especially compared with fast twitch skeletal muscle. But it uses between 10 and 300 times less ATP to maintain the same tension. So you get slow tension generation and slow tension release with greater efficiency and less investment of ATP. So the latch bridge mechanism maintains tetanus, which is resting muscle tone. So for example, your gut when you're alive is one length. It's about 2/3 longer in death because you lose smooth muscle tone in your intestine. Another example of smooth muscle tone is vasomotor tone. So your arteries are kept in a state of partial contraction such that if you have a spike in blood pressure, the smooth muscle can relax, or if you have a drop in blood pressure, the smooth muscle can contract to maintain your blood pressure at about 120/80-ish. So let's look at the schematic diagram now. So here we have calmodulin. Remember, this is calcium modulating protein. And calcium, which is going to come from the extracellular fluid or a variety of other locations. But let's just say it's coming from the ECF because that's simplest. So calcium plus calmodulin is capable of taking myosin kinase and activating it. So inactive myosin kinase can't do anything. Active myosin kinase can do something. So a kinase is any enzyme that adds a phosphate group to a particular molecule. And usually whatever molecule that is in the name. So myosin kinase means this enzyme adds a phosphate group to myosin specifically. So active myosin kinase is going to add a phosphate group from hydrolysis of the first ATP to inactive myosin. And once myosin is phosphorylated, it can bind the second ATP and bind with actin and produce the power stroke. So it makes sense that this is more complicated, or excuse me, that this takes longer than skeletal muscle because there are more steps. So what this looks like is we have a long fusiform cell. Here are the thin filaments attached to the dense bodies. Here's the thick filaments. This isn't a sarcomere though, because it's not a repeating pattern. And when calmodulin binds to calcium and activates myosin kinase, that creates a situation in which these thick filaments are capable of binding to actin, and you get contraction. But it twists up and bunches up the cell. So now it looks like you've sort of like stuffed a leg or a foot or something into a too small fishnet stocking. So we've got bulges. So it goes from being relaxed to contracted, just in a different way. Other types of muscle tissue include cardiac muscle. So cardiac muscle we will be discussing more in 232 when we discuss the cardiovascular system, and it's found only in the heart. So the cells are involuntary, but they are striated. So striated equals they have sarcomeres. And the cells are branched cylinders. You can see these guys have lots of branches, but they only have one nucleus instead of the many that skeletal muscle have. And we call these cardiocytes or cardiomyocytes. So cardiocyte means heart cell, cardiomyocyte means heart muscle cell. They are the same thing. So these guys are attached to each other by structures called intercalated discs, aka the question on the practical that everyone got wrong. Don't worry, you'll see these again. So intercalated discs are a combination of desmosomes, which are the rivet-like cell junctions that keep them from pulling apart, and gap junctions, which allow ion flow between adjacent cells. And that allows these cells to depolarize and rhythmically contract in an ordered way to produce your heartbeat. These cells are also autorhythmic, so there are populations of muscle cells in your heart called pacemaker cells that depolarize spontaneously and set the base rhythm of your heart, even without input from your vagus nerve. So cardiac muscle cells are shorter, thicker, and branched. And as I stated, electrical gap junctions allow cells to stimulate their neighbors. The mechanical junctions, including the desmosomes, keep the cardiomyocytes from pulling apart during contraction. The sarcoplasmic reticulum is present, but similar to smooth muscle, it's less developed. So it's not big and it doesn't come in big, robust terminal cisternae. It looks different from that of skeletal muscle. And the T-tubules, accordingly, are larger so that the cardiac muscle can rely on extracellular fluid, calcium, to power the excitation-contraction coupling. So this means that this is another reason why calcium in your blood is regulated closely, because your heart relies heavily on ECF calcium as opposed to stored calcium. Unfortunately, if you damage your heart muscle cells, say you have a coronary ischemic attack, which means one of your coronary blood vessels is blocked by plaque or a clot or some goober, the cells that die as a result of not getting oxygen are not replaced by other cardiomyocytes. They are replaced by fibrotic tissue, which isn't contractile. So the more ouchies you do to your heart, those can accumulate over time to produce a non-functional heart. So that's why people that have had several heart attacks need a heart replacement, because they can't replace what they lost. Your cardiac muscles rely on aerobic respiration almost exclusively. So very little of that lactic acid or phosphagen business. So they have huge mitochondria and lots of them that make them very resistant to fatigue, but that means that they're very vulnerable to interruptions in oxygen supply, which goes back to that analogy I made about a myocardial infarction. So more on that when we get to the cardiovascular system. Okay, so we're going to end with some clinical correlations, which hopefully will be useful to you. So muscular dystrophy is a hereditary group of diseases, so there's not only one kind. There's Duchenne's muscular dystrophy, and there are others as well. For whatever reason, it primarily is a disease of males, and this is not the most inclusive language I could possibly use. What I mean here is XY individuals, so there seems to be some gametic sex linkage related to muscular dystrophy. I apologize, I stole some of this verbiage from the stock PowerPoint. So it typically appears as a child begins to walk, and that's because you get skeletal muscles that tear because dystrophin is not linking the sarcolemma to the actin filaments. So when the muscle contracts, there isn't a concomitant constricture of the cell membrane, which means that every time you contract your sarcomere, you're accumulating little micro tears in the skeletal muscle fiber. So over time, you get tissue that atrophies and is replaced with fat. So you can see the muscle density in this individual is much diminished compared to the muscle density in this healthy individual. One of the first signs of muscular dystrophy is the toes begin to point as the calf muscles begin to constrict, and that's why it usually appears as child begins to walk. So your kid either doesn't walk or walks normally for a little bit and then begins to walk on their tippy toes as this process occurs. So these individuals rarely live past 20 years of age. It's a very sad disease, and it's really challenging for families. One weird version of muscular dystrophy is facioscapulohumeral, so that's face, shoulder, and upper arm. And this is muscular dystrophy that only affects the upper body, not the rest of the body. Myasthenia gravis is an autoimmune disorder. So in this one, it's not congenital or hereditary, but rather the individual's immune system, so their white blood cells and antibodies and such, attack things that are supposed to remain unattacked, in this case, the neuromuscular junction. So they bind to acetylcholine receptors and remove the receptors. So they treat acetylcholine receptors as a foreign invader to be destroyed. So with removal of these receptors, you get less and less sensitivity to acetylcholine. So there's nothing wrong with the neurons. They're still producing acetylcholine like normal, but the available receptors to receive the acetylcholine and excite the sarcolemma are going away. So you get progressive weakness and drooping of eyelids. You also get double vision as your eyes are no longer able to converge on the same thing because the rectus muscles of your eyes back in here are skeletal muscle. You get difficulty swallowing, weakness of limbs, and eventual respiratory failure if left untreated. So for whatever reason, this tends to be a disease of XX individuals between the ages of 20 and 40. Not sure why, still being worked on. There's probably a PhD in there for you if you want it. To treat this, you remove the thymus, just in case the thymus is still inappropriately producing immune cells into adulthood. Your thymus normally goes away after childhood. You can also use cholinesterase inhibitors, which increases the amount of leftover acetylcholine in the synaptic cleft, in combination with immunosuppressive agents, which prevent your white blood cells from making antibodies to attack the acetylcholine receptors. So cholinesterase inhibitors plus immunosuppressive agents can significantly slow down this disease. And that concludes the muscular system. So thanks for following along. Stay tuned for the next set of PowerPoints, which will be an introduction to the nervous system. So if you thought that action potential and membrane potential time was over, I have bad news for you. It will continue to be a thing. So not a bad idea to go review the videos I made about those. All right, toodles.